I'm not coming today.
Oh my God.
وسلم أستحق I had 10 minutes left on the other lecture.
Are you caught up?
Yeah,
You all caught up now?
I had like 10 minutes left. Man.
Damn. Until I
What is it, 5 minutes? 6? Yeah. That's the point.
That five and white.
Just split colors.
All right, so in the first 5 steps, nothing happened, right? No ATP was made. In fact, you actually invested 2 ATPs in this whole process. So the ATPs will be made in the remaining steps of glycolysis. And going from glucose to glyceraldehyde 3-phosphate, you are converting a 6-carbon sugar to 3-carbon sugar, so you get 2 of these. So every step after this will be multiplied by 2 because you are calculating based on 1 glucose molecule. So glyceraldehyde 3-phosphate will eventually be oxidized. So this aldehyde will be oxidized to COOH. So because the product will be a carboxylic acid, then this is a glyceric acid, so this is called 3-phosphoglycerate. Yes?
Sorry, just one question. What step is this exactly? 5 or 6?
6.
6.
So in this reaction, aldehyde is converted to the acid through another step in between. Now, in organic chemistry, we have learned that aldehydes are strong reducing agents. They can easily get oxidized. In fact, it is a task to keep it in the aldehyde state. If you open a bottle of aldehyde and then after maybe a few months, it will not be an aldehyde anymore. It gets oxidized from oxygen in the air. So in the organic lab, if you are given an unknown that is supposed to be an aldehyde and you don't see it's reacting as an aldehyde, a bit of the stockroom made the stock long time ago and it got oxidized. So it is difficult to oxidize, it is easy to oxidize aldehydes, and the cell actually does that in 2 steps. We have seen such examples before. Why does the cell take 2 steps to do such an easy reaction which should have been done in 1 step? Because it easily gets oxidized. And the reason, as I mentioned before, always has to do with energy utilization. That is the most important part, that you will be using the energy of oxidation. Whenever you oxidize any organic molecule, that releases energy. So this oxidation will also release a lot of energy, and that energy is used to make ATP. So you want to make ATP in going from the aldehyde to the carboxylic acid, and for that, it will go through an intermediate step. And what is that intermediate step? You put a phosphate, because remember, if you oxidize it, it releases energy, and the oxidizing agent cannot be oxygen, because we are talking about anaerobic pathway, right? The whole glycolysis pathway is anaerobic. There is no oxygen in the cytoplasm. And so you have to use a different oxidizing agent, and that is NAD. As I mentioned before, NAD is always written with a plus, right? NAD plus. That's very confusing because NAD actually has negative charges. NAD is negatively charged. It's written as a plus just to focus on one carbon that has a plus charge, but it also has phosphates, right? Nicotine, adenine, dinucleotide. The dinucleotide says it has 2 phosphates. That means it has all the phosphates have negative charges. So overall, the molecule actually has negative charges, but it's written always as NAD plus. Just to focus on that particular carbon in the molecule that has the plus charge. So NAD is the oxidizing agent, so it will get converted to NADH. So if you want, as I mentioned, usually these reactions are written above the arrow as a reaction. So instead of writing just NAD, you can write it this way, that NAD gets converted to NADH. So that's another reaction that happens at the same time. So when you oxidize that, that will release a lot of energy, and that energy is used to form a bond with inorganic phosphate. So phosphate ion in solution will be used to form a bond with this carbon. So instead of getting the carboxylic acid, you get so I sometimes write the phosphate with all the oxygen, sometimes I don't. With the circle, it means that it has the 4 oxygens also. So this will be then, if this is glycerate, this is also glycerate with a phosphate in there. So there are 2 phosphates. It is at carbon 1 and carbon 3, so this is bis phosphoglycerate. So 1,3 bis phosphoglycerate. And the reason it went through this intermediate is because you want to conserve the energy of the oxidation in this bond. So you have a phosphate bond here, that is a high energy compound, and that will be used to make ATP. So if you look in the list of all the high energy compounds that I gave you, on top of the list is phosphoenol pyruvate, and right after that is 1,3-bisphosphoglycerate. That is one of the high energy compounds, and so its function is to make ATP. So this will be used to make ATP, and that means in this next reaction, ADP will be converted to ATP. And you get 3-phosphoglycerate plus ATP. So this is the first reaction in which you are actually making ATP. Previous reactions we were investing ATP, and because, as I said, that you have 2 of these, you are actually making 2 ATPs for glucose here. So what will be the name of this one? As I said before, anything that puts a phosphate on a molecule, that is called a kinase. It doesn't matter whether the phosphate is coming from ATP or used to make ATP. It is a transfer of a phosphate, so the enzyme is called kinase. So this will be phosphoglycerate kinase. So that's the name of the enzyme. It's forming phosphoglycerate and adding ATP to phosphate to ATP, so it's a kinase. So here you make 2 ATPs, and once again you are going through this intermediate because energy of oxidation is conserved in that bond. Yes?
What's the name for the first enzyme?
Oh, for the first enzyme, yes. So you are doing, you're making, taking this one, 3-phosphoglycerate. And you are doing an oxidation reaction in which you are removing 2 hydrogens. So this is called a dehydrogenase. 3-phosphoglycerate dehydrogenase. And that uses NAD as the oxidizing agent. Remember, for all anaerobic reactions, oxidizing agent cannot be oxygen. So it has to be usually NAD. And in some cases, if it is FAD, always remember that FAD is a prosthetic group. It cannot give you ATP later. So if any enzyme uses a FAD, in the next step, there will be another enzyme that will convert the FADH2 back to FAD and then give you NADH. So NAD is somehow involved either directly in that step or in a later step. It has to be NAD. All right, so this is where you make 2 ATPs, and in the next step, you are going to move this phosphate from 3 position to 2 position. So this was 3-phosphoglycerate, so this is 2-phosphoglycerate. And if you are moving a phosphate from one position to another, as I mentioned before, the name of the enzyme will be what? What transferase? No. It is a mutase. It is called a mutase. As opposed to moving a carbonyl group or OH group. Whenever you are moving a OH group from one position to another, for example, glucose to fructose, those are called isomerases. And if you are moving a phosphate from one to another, all those enzymes are called mutases. So this will be then phosphoglycerate mutase. One point that I mentioned before, one property of mutases is that the phosphate that is being transferred from one position to another, it is that exactly same phosphorus, the oxygen that is transferred. Unlike moving a OH from one position to another, where the OH is actually first lost as water to form the enol, if there was a carbonyl group, or form a double bond if there is no carbonyl group, and then another water molecule is added. So first you lose a molecule of water to form an alkene, and then you add a molecule of water to form the alcohol back again at a different position. So it is not exactly the same oxygen molecule that goes to a carbon. And the reason is because all reactions are happening in water. Water is present at 55 molar concentration. If you are transferring a phosphate, that is present in millimolar concentration. So if the phosphate will leave the molecule, it is not going to come back. It is a very diluted solution. It's very difficult for the enzyme to get another phosphate back and put on the next carbon. So whenever it is a phosphate that is being transferred, the phosphate never leaves the enzyme. Actually, all these mutases come with a phosphate. So one of the amino acid at the active site has a phosphate, and this one is transferred to another amino acid, and this amino acid transfers its phosphate back to this other position. So basically, you have the active site with the phosphate here, and whatever molecule you are transferring the phosphate from. So this phosphate will go to another amino acid, and then, this phosphate first goes here, so that gives you this molecule with 2 phosphates. So one phosphate is already with the enzyme, this gives you 2 phosphates, and then this goes back there, and that gives you the phosphate at a different position. Alright, so your enzyme gets back the phosphate that it had, but now the phosphate is at a different position. So phosphate goes from the substrate to the enzyme came with the phosphate, and that phosphate goes to the substrate. So the enzyme always has a phosphate there. That phosphate is never lost into the medium, because if it does, then the enzyme cannot find it anymore. It's too diluted. And that is true for all mutases. So as a general diagram I drew here, there are many mutases we will see, and they all work this way. Alright, so as I said before, every reaction has a purpose. As we saw here, the purpose of oxidizing, going another step, is to make that ATP. And then what is the purpose of moving the phosphate from 2 position to 3 position? And that is, you can see in the next step, where you are going to lose a molecule of water. So this OH, and there is a hydrogen here that is lost, and you get a molecule of water, and that gives you So you get the enol form of the phosphate, right? Remember, if you have a MOS structure like this, with a OH, this is called an enol. And instead of A8, you have a phosphate, so this is a enol phosphate, and this was your phosphoenol pyruvate. So phosphoenol pyruvate is a high energy compound, and we have seen before why it is a high energy compound, because it is stuck in the enol form, and it is used to make ATP. It is one of the highest energy molecules that has the highest phosphoryl transfer potential, and that was the reason for moving the phosphate from 3 to 2, so that you can get the enol phosphate. So obviously in the next step, and all these reactions I'm writing in one direction, but I already told you there are only 3 irreversible steps, steps 1, 7, and 10. So these are actually all reversible steps, and then in the next step it is an irreversible step where this phosphate will be transferred to ATP. So you get pyruvate. So this is pyruvate from phosphoenol pyruvate. When that phosphate comes off, it becomes the enol, which is unstable, so it immediately converts to the ketone and gives you pyruvate. And in the process, you are making an ATP, and the enzyme, of course, is a kinase, because you are making ATP and the product is pyruvate, so the name is pyruvate kinase. So that's the end product of glycolysis, but as I said before, the pathways are named by us. The cell doesn't know there is a pathway. It doesn't know that it has to stop at pyruvate. It doesn't actually, you need to take care of this pyruvate after this. So this is the second step where you are making ATP and you are multiplying that by 2, so that makes it 2 ATPs here and 2 ATPs there, so you are making total of 4 ATP. And you had invested 2 before, so your net result is 2 ATP per glucose in glycolysis. Alright, so as I mentioned before, we will be studying many pathways, and there will be some reversible, some irreversible steps, and I may forget or may not even mention that there is regulation. So you should know that all pathways are regulated, and they are regulated at the irreversible steps. So just even though I don't mention it all the time, let's talk about it now. Which steps will be regulated? Those that are irreversible. The first one was putting a phosphate on glucose. Second one was putting a phosphate on fructose to give you fructose 1,3-bisphosphate. So those are the 2 steps regulated, and then this is the last step that is also irreversible, so pyruvate kinase is regulated. And the regulation is usually by feedback inhibition. So the end product of glycolysis that the cell wants is ATP, so ATP will always inhibit these earlier steps. The immediate product of a reaction is also an end product that can inhibit that step. For example, glucose to glucose phosphate, that means glucose phosphate will itself inhibit that enzyme. So immediate end product and end product of the whole pathway both can be feedback inhibitors. The reversible steps are usually not regulated, so they don't have any feedback inhibition. So ATP is a common inhibitor. Then there are other inhibitors. Some of them are also regulated by covalent modifications. So I spoke about the general method of regulation. Sometimes they apply to all, sometimes they apply to some. But we will not be talking about regulation of everything unless there is something specific to mention. All right, so once you make the pyruvate, what happens to it? This is all in the anaerobic pathway. What happens to pyruvate is that it goes to the aerobic cycle, which we will talk in a different chapter. But sometimes there are other reactions that also happen with the pyruvate. For example, in humans, the pyruvate can be converted to lactate. Right? And why does that happen? It happens in the anaerobic. It's happening. All is happening in the cytoplasm where there is no oxygen. Other species, like for example, some bacteria, yeast, they undergo fermentation, and in that process they convert their pyruvate to acetaldehyde and then to ethanol. So everyone converts it to something, but they don't need that product. Yeast don't need that alcohol. We don't need the lactate. We have no use for the lactate. Yeast also has no use for the ethanol, but they do it. So why is pyruvate converted to lactate? And that happens because in the absence of oxygen, you need to make more and more ATP. And so pyruvate is converted to lactate, and people get muscle cramps because you are exercising too much. And you will often find this misinformation or myth, especially if you are relying on Mr. Google for all your information. You will get this myth that lactic acid gives you that muscle ache, which is not true, right? Lactic acid doesn't give you that muscle ache. As a experienced organic chemist, you know that here is pyruvic acid, which we always call pyruvate, and here is lactic acid. This is lactic acid, this is pyruvic acid. Which is the stronger acid? Yes? Which one is stronger?
Lactate.
Why can you not see the board? We have straight line. Alright, which one is the stronger acid of these two? Pyruvic acid, why?
Double bond.
Because of the double bond. It pulls more electron from this this hydrogen, right? So if you write this way, and this one this way. So the double bond oxygen pulls more electron from this one, then that makes it a stronger acid. So pyruvic acid is a stronger acid, and we don't call any of these carboxylic acids as acids. They are all ates, like pyruvate and lactate. But the general public calls it a lactic acid, and pyruvate they have never heard of. So this lactic acid, whenever you hear the word acid, it must be very bad. Right, so lactic acid causes muscle ache, but actually it was pyruvate before, which is a stronger acid. But that didn't give you muscle ache. Why does the lactic acid give you muscle ache? In fact, that has nothing to do with the pyruvic acid or the lactic acid. So what does it have to do with? It has to do with the fact that you are doing exercise. You are consuming ATP, and ATP is converted to ADP. So when you are doing exercise, ATP is hydrolyzed, which is the wrong statement to make. I'll explain in a minute. So ATP converted to ADP plus phosphate plus, if you balance the equation, there will be acid. You have to balance the equation with the proton. So I mentioned before that high energy compounds are never hydrolyzed, but I wrote a reaction here that it is hydrolyzed, which is not true. It is not hydrolyzed, but it goes through several steps. The phosphate is transferred to something else, and then eventually it will give you free phosphate. So your purpose is to contract muscles. That's your exercise. You are doing mechanical work, and that's where the phosphate transfers to another molecule and then to another, and that's how you do your exercise. But eventually the net result is that it will be converted to ADP phosphate, and you are releasing a proton just to balance that equation, and that is the proton that gives you muscle ache. More exercise you do, more acid it forms, and the pH goes down. And because the pH goes down, that gives you muscle ache. It has nothing to do with the lactic acid. In fact, it is not even lactic acid, it is a lactate ion. All right, so I'm going to erase the top part.
This? The biggest tangent is gonna be what's on the section of
Can you explain why it's the proton that cause the muscle pain?
Because it lowers the pH.
I want to see that.
PH goes below normal, and that's the problem with muscle pain. So don't rely on the surgical for your information. All right, so the question is, why does this reaction take place? Why do we humans convert it to lactate, and why other organisms do something to it to convert it to something else? Why does that happen? Why does this reaction happen? This is you're converting this alcohol to this ketone to an alcohol, and so that is a reduction step, and that will need a reducing agent, and that reducing agent will be NADH. NADH will be converted to NAD. And the reaction is happening because of this reaction of forming NAD. Now remember, in glycolysis, I just erased that reaction where glyceraldehyde, 3-phosphoglycerate is oxidized with NAD to give you the 1,3-bisphosphoglycerate. So that reaction, 3-phosphoglycerate dehydrogenase, glyceraldehyde dehydrogenase, that requires NAD. So in that reaction, I'm not writing that again, but this was one of the. This is the only reaction in the glycolysis that uses NAD as the oxidizing agent. And because this is happening in the cytoplasm, you have only a limited amount of NAD in the cytoplasm. And if you are doing glycolysis, all that NAD is used up. And if you are doing exercise without getting enough oxygen, that means you need to do the glycolysis again and again. For yeast, they have no other option, they have to do it again and again because they are doing fermentation in lack of oxygen. And so you need more NADH. All your NADH is, you need more NAD, but all your NAD is used up in this reaction. So you need to get back your NAD, and that's the reason you need to convert the pyruvate to lactate. So this NAD then continues your glycolysis repeatedly. So only you can do your glycolysis only a few times. After that, you are relying on oxygen to take care of the pyruvate. But if you want to keep doing it again and again, you must get back your NAD. And this is the reason this reaction happens. In humans, we convert to lactate. In others, other reactions. But the whole purpose is to get back that NADs to repeatedly do more glycolysis. Because glycolysis, remember, gives you only 2 ATPs, whereas one glucose ultimately will give you 32 ATPs. So you are not getting the benefit of the remaining 30, you are just relying on that 2 ATPs every time. All right, so what happens to this lactate then? After giving you the muscle ache, or not, if you haven't done that much exercise, you don't have that, but eventually you are forming this lactate, and then this lactate will go from the muscles. So this is in the muscles, then it goes to blood, and then it goes to the liver. And in the liver, this opposite reaction takes place, where lactate is converted back to pyruvate. So as I said, we have no use for the lactate. And the yeast also has no use for the ethanol. So what do we do with the lactate? We convert it back to pyruvate in the liver. The liver is an organ that works for the rest of the body. There are many reactions happening in the liver, and this is one of them, to convert the lactate back to pyruvate. There is another place that makes a lot of lactate, although they are not doing much exercise, but they make a lot of lactate that again goes into the blood, and then that goes to the liver, and then it is oxidized to make pyruvate. And that happens in the red blood cells, erythrocyte In the erythrocytes, you have red blood cells that cannot do oxidative reactions. What is the function of red blood cells? To transport oxygen. For that, it needs hemoglobin. To make that transport process more efficient, the red blood cells pack in as much hemoglobin as possible. Because more hemoglobin you pack in, more oxygen you can transfer. So red blood cells are basically packed with hemoglobin. There is no space for anything else. So it doesn't have most of the enzymes. It doesn't even have those genes. It has less DNA there. So it doesn't have the genes for citric acid cycle or any other cycle that gives you more energy. So it relies entirely on glycolysis for all its needs. And that means they will have a lot of pyruvate. And what will happen to that pyruvate? Same as here, it has to be converted to lactate to do more glycolysis. So red blood cells will release a lot of lactate into the blood that again goes to the liver and then converted to pyruvate. The name of the enzyme I said before is the same as the one that is in the muscles, lactate dehydrogenase. So the name doesn't tell you which direction the reaction goes. It can go in either direction. All right, so as I said, your exam on Q quiz, which is a short quiz, will be on glycolysis. But there is one more thing from glycolysis to do, and that is about a branch in this pathway in the red blood cells. So red blood cells, you will convert glucose through all these steps to give you eventually (3-phosphoglycerate). All right, so this is 1,3-bisphosphoglycerate, which you can say is 1,3-BPG. And then it will, of course, give you the rest of the steps of pyruvate. So this is your glycolysis step. But there is a branch that takes place in the red blood cells only, and that is to make another compound that is very important for oxygen transport, and which we already saw in the previous chapter. What is that molecule that regulates oxygen transport, that inhibits binding of oxygen? That was the 2,3-BPG, right? So this is 1,3-BPG, and that is 2,3-BPG. So even though this is the main pathway that is giving energy to the red blood cells, some of the molecules of 1,3-BPG will be converted to 2,3-BPG. So there will be a phosphate here and a phosphate here. So this is 2,3-BPG. When you make ATP in the next step, remember in this step we were making ATP. So ATP makes ATP in the very next step of glycolysis. By transferring a phosphate to ADP, you make ATP, which are ATPase. We had the equation before. The 1,3-BPG has two phosphates there. Which is the phosphate that will be transferred to the ADP? Out of the two, which one will be transferred to ADP?
The one.
One, which one?
The one with the double bond next to it. The one oxygen.
The top one,
Yeah.
carbon 1. So this phosphate will be transferred, not this one. And why is that? Because of what kind of linkage? What is the linkage in this one? This is an ester. OH with a phosphoric acid, that is an ester linkage. And this is a acid anhydride linkage. Right? So this linkage is acid anhydride. And acid anhydrides are unstable. They hydrolyze more easily. They have a higher energy of hydrolysis. And that is the definition of high energy compound. So that is the one that will be transferred to make ATP, not the phosphate at the third position. When you move the ATP, when you move the phosphate from here to here, you are getting 2,3-BPG. This is also an ester linkage, not an acid anhydride linkage. Right? So that means the excess energy in this one will be lost as heat energy. And it will not, anything that is lost, of course, you cannot get it back again, so you cannot really make any ATP from that. It's already gone. Right? And this inhibits oxygen binding, like when you go up the mountains, your BPG level increases, and when you come back, your BPG level decreases again. What happens to the extra BPG? It will be hydrolyzed. So this will be converted back to 3-phosphoglycerate. That means this phosphate bond will be simply hydrolyzed because it doesn't have enough energy to make ATP. So basically in this branch, you were making 2 ATP by glycolysis. If you go this way, you are making 1 ATP because one of these phosphate is simply hydrolyzed. So it first went from position 1 to 2, so lost some energy there, and then from 2 to 3, there is not enough energy to make ATP, so it is simply hydrolyzed. So sometimes high energy compounds are hydrolyzed if there isn't enough energy to make ATP, but hydrolysis will make the reaction irreversible, and that's how this reaction eventually goes to the 3-phosphoglycerate. So anything that is shunted this way gives you 1 less ATP, but that doesn't matter. It's because that's only a minor pathway. The major pathway is your glycolysis. Only when you need more ATP, 2,3-BPG, some of it will be shunted to that extra branch. All right, and if it goes that way, then you don't make that 1 extra ATP. And that happens in the red blood cells only. You don't need 2,3-BPG anywhere else. All right, so that's the last thing in glycolysis. As I said, your quiz will be up to this point. 4 main topics, 2 questions each. And then your midterm is coming up after that. And the midterm will end at all the fermentation steps, that means all non-anaerobic steps. We have at least 2 more things to discuss about that. One is breakdown and synthesis of glycogen, and then gluconeogenesis is another pathway, and then pentose phosphate pathway. So these 2 pathways we will discuss briefly before the midterm. Yes?
I have a quick question. How many questions is going to be on the midterm?
I have no idea. I haven't written the exam.
Is it on paper?
Is the exam on paper or is it going to be on?
No, it's exam is on exam software. And don't blame me for it, I don't decide that. It is already decided by pharmacy. And the reason they have it this way is because in exam soft you cannot go back on the questions.
What's an option that is up to you like that?
I have not given any option. So whatever examsoft does, so you cannot go back on the questions, and that's how they want it. Because that's how your board exams are, and they want the midterm to be that. Yes?
Is it multiple choice or is it like?
Multiple choice.
All right, good.
So all multiple choice, you don't have to draw something.
You at least get a 25.
Not me.
ABC.
We have backtracking for pharmaceutics.
Good thing that came well.
They are greater people. I don't have that option. I'm asked to leave it up.